Welcome. Hope you had a nice night. Well, welcome to class. This is CRP 305, Genetics and Breeding, Facilitation, and I'm still your facilitator, not changed. ALAWODE Yetunde is my name. So today we'll be talking about hereditability. Heritability. Heritability is the topic today. But before we continue, I saw your assignments. And I made necessary corrections. I'm not going to correct it on this platform. I need you to submit to me individually so that I will see what you can do by yourself. Like I said, I'll just set my questions. I don't know which one DEA will pick. So that is why I'm trying to make sure that I touch everything that I can touch before the exams. So please take it seriously. If I tell you to do assignment, do it. Let me see how well you understand the class. So it will not be as if I'm talking to myself or I'm the only one. So that's why I said, so I'm not going to come and do it for you here. I want you to submit to me individually. And for those that are submitting to my email, I prefer that you submit it to my WhatsApp. I can't make correction on your email, but I can make corrections on the WhatsApp chat. So please, let's do the necessary thing that we are supposed to do. So you're welcome again to class. And before we continue, I want us to see something together because I think I'm satisfied with what you have done for those of you that have submitted, and you know how to do the calculated chi-square. So it's the tabulated that is remaining, because you know that you are going to compare the two results. You have your calculated, that's the one that you did on paper. Then you will now go and check the tabulated, the one on the table, on the chi-square table. So you will now compare the two results, the calculated and the tabulated, to know if it is significant. So to be significant, like I've written here, to be significant, the calculated must be greater than the tabulated. Hope you understand. The one you have calculated, it has to be greater than the one on the table to be significant. Okay? But if it is not significant, what you mean by if it is not significant, that means that the one in the table is greater than the one calculated. In that case, then it is not significant. So if it is not significant, then you should not disturb yourself. That's what they are trying to say. You shouldn't disturb yourself. That means that the result is as good as the Mendelian ratio of 3:1. You know that the Mendelian ratio is the one that you should expect. But when you did your observation, you now saw that there were differences. So you want to compare the differences. Is this something you should be worried about? That is what this chi-square is all about. Is this something you should be worried about? So if it is not significant, then that means that your Mendelian ratio is still good for you to use. Hope you understand that. So from the previous example that we did in the last class, let us go and check the tabulated value. Now, let's check the table. Let me share the chi-square table. This chi-square table is the same everywhere. Everywhere in the world is the same. So there's nothing like a chi-square table for something or something. Let me use the chi-square table that you were given in your course material. So I'm sharing the chi-square table on your course material now. This is what it looks like. Hope you can see it. This is what the chi-square table on your course material looks like. So what you need to check the tabulated value now is this degree of freedom. You can see degree of freedom on this side. Hope you are following. You can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 on this side. That's the degree of freedom. And you can see the probability on this side. The probability is horizontal and the degree of freedom is vertical. I don't expect you to cram this table. It's not possible. As a matter of fact, I don't even want you to take any sheet of paper to the exam hall because it could implicate you. So I'm just going to teach you how to look at it. If it comes out in the exam, I will give you this value by myself. I will give you the tabulated value. But I just want to teach you how you can look at it. I will give you the tabulated value, and I want you to compare it to the calculated value that you have calculated so that you can compare both results. But so that you will know how to look at it, you need this degree of freedom vertically and the probability horizontally. Now in agric science, the probability level we normally use is 0.050. This probability, this one, is what we normally use in agriculture. In other sciences like medical sciences, where you deal with human life, they use this 0.99 probability because you cannot afford to be playing with life the way you are playing with plants. So this is the level of probability that we normally use in agric, 0.050. Now, in the case of two classes, for those of you that were in the class last week, in the case of two classes, the Mendelian class, ratio 3:1. 3:1 is two classes. You know, you did class of 3, you did class of 1. So that is two classes. Your degree of freedom will be 1. That means that whatever classes you are using, you subtract 1 from it. 2 classes minus 1 is 1. So your degree of freedom in that case of the 3:1 ratio is 1. So what you are going to do is that you are going to check your degree of freedom under the probability level of 0.05. So if you trace it down like this to this 0.05, the value that you will get under is 3.84. That is your tabulated value. Hope it is clear. You all understand? I need someone to answer me.
Yes, we understand.
Okay. So that is how it is. You will trace this degree of freedom to the probability level. This value that is directly under it is your tabulated value. So you have your tabulated, you have your calculated. You compare. If the tabulated, like I said in the slide here, that if your calculated is greater than your tabulated, then the result is significant. That means it does not follow the Mendelian ratio. That means there is a problem with your experiments. But if the tabulated is greater than the calculated, then it is not significant. It's not something you should worry yourself about. That means that the Mendelian ratio is still in play. So for the case, in the case of the 9:3:3:1 ratio, that is how many classes now? Who can tell me?
4 classes.
4 classes. So the degree of freedom will be what?
3.
3. The degree
Yes.
of freedom will be 3. So this is the
Yeah.
table now. What will the calculated, the tabulated value be?
7.81.
Good. All you need to do is to trace this degree of freedom down to where this 0.05 is. So the value that is underneath it here is 7.81. That is your tabulated value. So what I want you to do now is that you have your calculated, you know your tabulated. I want you to go and take that result that you submitted to me. Go and compare it and give me feedback if it is significant or not, and what you should do. Hope you understand. That is clear enough. The result, for those of you that have submitted your assignment to me, go and compare it to the tabulated value. You have this table inside your course material. You have it in your course material. So compare it and send me the feedback. So that is that's just a digression. So today's topic itself is heritability. Like I said, let me share screen again. So heritability. So why is it that when you take a corn cob, for example, and you want to plant all the seeds that are on that maize crop, you plant everything in the ground, the same rainfall is the one falling on all of them. The same fertilizer is the one you are giving all of them. But at the end of the day, you see that some are tall, some are short, some are in between, all manner of phenotypes. It's because of this heritability. Okay, so this heritability is a measure of the contribution of genotype to the total phenotype variability. Remember, in the beginning of this class, I gave you a simple formula. I said the phenotype, that's what you can see, is as a result of the environmental factors, the genotype, and interaction of genotype by environment factor. So that heritability now, this heritability is that portion of the G, that's the portion of the genotype, that thing that you collected from your parents that is making you to be taller than the rest of your siblings, even though you are eating the same food, you are sleeping in the same house, you are doing the same sun is the one shining on you, the same rain is the one falling on you, everything is the same. But at the end of the day, you are now taller than the rest of your siblings. So that portion, that genotypic portion, is what you inherited from your parents. Okay, so that is what heritability is. Is the measure of the contribution of the genotype to the total phenotypic variability. Okay, and it's also the portion of the total variability that can be inherited. Like I said, you normally inherit it from your parents. So also the crops inherit traits from their parents. So there are 2 types of this heritability. We have the broad sense heritability and the narrow sense heritability. So in the case of this broad sense heritability now, it is represented with capital H. You can see it here. capital H squared. That is how you, that's the symbol for broad sense heritability. And what it does is that it captures the portion of the phenotypic variation that is due to genetic values that may include effects due to dominance and epistasis. If you remember, I told you the reasons why this Mendelian ratio, in some cases, the reason why it is not accurate, like we did before we did a chi-square, I told you there is a ratio that you expect, but there's a ratio that is observed. So why is there why is the difference there? Why are there discrepancies here and there? Is it because of this dominance and epistasis theory? So that portion that is due to dominance and epistasis, you know, we are talking of the genetic portion now. Out of the formula of VPH is equal to VG plus VE plus VG, like you have here. This VG portion can be further divided into dominance and epistasis. So the part that affects that genotype that is due to dominance and epistasis is the broad sense heritability, and it has its formula. This is the formula. I don't want you to mix up the formula. So the formula, like I told you, it is capital H squared. The formula is VG all over VPH. There are simple formulas that you can know. There are simple formula you can know. So the VG is the genotypic variance. VPH is the phenotypic variance. And like you know from that formula that I gave you in the first class, that VPH is equal to VG plus VE plus VGE. These are simple things, so they are not complex at all if you just follow your mind. So this is the formula for broad sense heritability. In the case of the narrow sense, okay, that narrow sense heritability, it captures only that portion of the genetic variation that is additive, that is due to additive values. So we have the additive, the parts of the genetic variance. We have the additive portion, we have the dominance portion, then we have the epistatic portion. There are three portions that are responsible for the genetic variation. So out of these three portions, the dominance and epistatic portion is due to broad sense heritability, while the additive portion of that genetic part is due to narrow sense heritability. And narrow sense heritability is represented by small h. It's small h, and the formula is VA. This A is due to additive genes all over VPH. Okay, and in this case, the VPH is due to genetic variation plus environmental variation plus the interaction of the additive portion, because it's narrow sense. That's why you have VI here. VI due to interaction or epistasis. So we have your VG here as additive variance plus dominance variance plus the interactive variance. This is the formula for your VG. All these formula you can cram it or whatever. There are simple things you should know, but just make sure you know it. Just make sure you know it. Because like I said, you can never tell. Maybe it is this question that will come out in the exam or not. I don't know. It's in the hand of DE. But me, I want to satisfy my conscience and teach you everything that is in the material. So I listed the meaning of all this over here. You have VPH as phenotypic variance, VG as genotypic variance, VE as environmental variance, VI as epistasis or interaction variance, VA as additive variance, and VD as dominance variance. This is the meaning of all those things you are seeing here. So, how do you compute heritability value? Okay, the method of using what okay, the value of heritability depends on what?
Good morning.
How small or big? Are you talking, Oyedoku Olashade? You want to say something?
No, man.
Okay. Please mute yourself. Okay, so the heritability value, the value that you get after you calculate your heritability, depends on three things: the method of computation, the estimate of genetic variance, and the population from which heritability is calculated. So these are the three things that depend on the heritability value. Meanwhile, the estimate of genetic variance should be free from G by E interaction. G by E simply means genotype by environment action. That means that when you are calculating your genetic variance, separate it from environmental variance or the interaction of the environment on the genotype, if you want your result to be accurate. Now, I'm just teaching you these things. These days, if you want to do all these things, you have to subject your data to analysis. That is what analysis is all about. You cannot be doing it one by one. It's going to take a lot of time, but you have to know how these people came about this result. If you do your project now, when you get your 500 level, we do what is called analysis of data. You collect your data and then you subject it to analysis. The computer will do everything for you these days, but we just have to let you know how it is being done before the computer runs it. So that is the importance of everything we are doing. So you should be separated. So how do you estimate the genetic? How do you separate the genetic from the environment? You have to do that by growing your crops in different locations and by growing in different. If you do a G by E analysis, you may not understand this now until you get to your final year when you're doing your project. There's what is called G by E interaction. What you want to do is that you want to plant the genotype, you want to plant your seeds in different environments. How does it perform in the east? How does it perform in the north? How does it perform in the south? You collect all your results together and then you run it, you run the analysis. So you want to know the performance in different locations as well as the performance over a period of years, because you know that the climate used to change. So you want to know the effect of the environment on that particular crop. So you are planting it over the years because there are different climatic change here and there, and then you are planting them in different locations. So that is how the heritability is achieved. And which one is bigger? Which one is bigger? You can guess from what I've explained so far. The heritability is the genetic portion of the phenotype of the individual, the genetic portion. And I said this genetic portion can be split into three. There's the dominance, there's the additive, and there's the epistasis. And I said that the broad sense heritability, we calculate the epistasis as well as the dominance effect. So it's calculating two out of the three. And the narrow sense is only calculating the additive portion of the genetic inheritance. So from what I said, you should know that the broad sense should be bigger in actual sense than the narrow sense heritability. So broad sense should be bigger. That's the explanation here. Now, this is example 1. You people say I've come with mathematics again, but Then you just have to know it. They are simple things, like I said. They are all simple things. So this question says, calculate the narrow sense and broad sense heritability if the following estimates were found for a population of maize plants. So you have a population of maize plants, and then you have the additive variance to be 20 units square. You have the dominant variance to be 10 units square. There should be square here. Sorry about that. You have the environmental variance to be 35 units square. Then the combination interaction of genetic and environment is 10 units square, and the interaction of the epistasis interaction is 5 units square. So they want you to calculate the broad and narrow sense heritability. So let's start with the broad sense heritability. That's the capital H. And you have the formula to be VG over VPH from the earlier slide that I showed you. Then you have your VG is in the early slide too, to be VA plus VG plus VI. So you just substitute the values from your question. You have VA, you have VD, you have VI. When you add it together, you get 35 units square. And for your VPH, you know the formula also. So this VG that you got now is what you will use. Then you have your VE in your question, you have a VG in the question. So that makes 80 units square. So all you have to do is just to put 35 all over 80. And for the broad sense, you have 35 over 80 here. You have 0.4375. And heritability is always reported in percentage. So you have to convert whatever result you have to percentage. So you have 43.75% over here. In the case of the narrow sense heritability, this is the formula. Narrow sense is small letter h squared. The formula is VA all over VPH. You have your VA, your question. You have your VPH from what you calculated in broad sense. That's 80, okay? So you have your 20 over 80, which is the same as 1 over 4 or 25%. So your broad sense heritability to this question now is 43.75%, and the narrow sense is 25%. Any question? Let me hear from you. Any question?
No question. No question for now.
Please, I want to understand the meaning of pH, the VPH.
Can somebody answer her? What is VPH?
VPH. VPH is the phenotypic variation.
Good. So do you understand that now? That's the variation that is due to phenotype, phenotypic variation of phenotypic variance. Okay, so that is what VPH is. You have your broad sense and narrow sense heritability. So this is another assignment. Please do your assignments. Let me see them individually. Like I said, I'm not going to come and solve anything for you in class. I want you to solve it by yourself, send it to me, let me correct it individually. I will take time. If you notice it, for those of you that even submit as at midnight, I see it, but I'm awake. I see it and I correct it immediately as soon as I have time. Please submit it. Let me check. So this is an assignment. You have your VPH, 50 units square. You have your additive variance, 10 units square. You have your dominant variance, 5 units square. You have your genotypic variance to be 20 units squared, then you have the interaction of additive to be 5 units squared. It should be square. Sorry, it's not subscript, it should be superscript. The question is calculate both broad and narrow sense heritability values. So please do it and submit. Then we'll be talking of another topic today, selection response. So selection is the process by which certain individuals are allowed to contribute more towards the progeny at the expense of others. Plant breeders do this thing a lot. We select. As a breeder, you should have eyes for selection. Just like some of you men, permit me to say, you have your preferences in your females that you want to choose. Those of you that want to marry, know who you want to marry. Apart from the character, apart from the fact that the person is God-fearing or whatever, you have physical attributes that you want in your woman. Some of you like women that are fair in complexion, some like women that are dark, some like women that are busty, some like women that, whatever, you know it, you have your preference. So also breeders do when it comes to plants. The plants that will produce high quality, that is what you want. So when you go to the field, you select so that you will be able to know the seeds that you will use for your next growing season. So you are allowing that plant to release whatever it has, contribute towards the progeny, because it's what you select that you will get as your progeny. So that is what selection is all about. It's the process by which certain individuals are allowed to contribute more towards the progeny at the expense of others, because you will see some others, you won't even go near it. You say, this is not what I want in my crop. This is what I want in my crop. So you will select based on that. You as the breeder know what you are selecting for. Is it tall plant you want? Sorghum is tall. Sorghum, for those of you that know, when you plant sorghum, sorghum is tall. But breeders don't want tall sorghum because it is very difficult to harvest. So they go for short varieties that you harvest, because what you are harvesting is on top, the panicle. So they want short varieties that you will harvest and it will be fast. So it's not in all cases that you prefer tall crops. Those that plant tall sorghum, they are those that want to use the stalk to build maybe fences or whatever. But in the actual sense of it, a breeder will go for a short variety sorghum plant. How I now, so let me just not to close. So depending on what you are selecting for, that is what you as a breeder should do. So that is about selection. And the well-accepted features of selection, there are features of selection, is that selection is effective only if heritable variation is present. Yes. So selection is very effective if what you are selecting for is something that can be inherited. If the trait is inherited, then fine, it is effective. But if it is due to environmental factors, then that means that if that thing is not present in the environment, then you cannot get the result you are looking for. For example, some plants, if it is the color of the flower you are looking for, some plants will produce certain color when they have enough sunlight. if it is the color you are looking for. So if that sunlight is not present, then that means you can't get that color. That kind of selection cannot be effective. But if the what you're selecting for is inherited, by inherited, then it is very, very effective. That means that all you have to do is just be looking for the parents that have that trait that you want. And then selection does not act on the gene itself, but acts on the genotype through the phenotype. I've explained this so many times that the phenotype gets is as a result of the genotype and the environment. So the gene cannot work on its own. It has to work through the phenotype. And selection itself does not create new genes, but genotypes, but influences the frequency. So that is why most people used to confuse genotypic ratio with phenotypic ratio. Phenotype is what you can see, like I always say, but the genotype is there. You cannot see it. You can only represent it with those letters that we used to use when we were doing monohybrid and dihybrid crosses. So selection does not create new genes, but they manipulate their genes so that they can improve or increase the frequency of phenotype. Now there's another short calculation here, calculation of genetic advance. Don't worry, after this class, I don't think there's any other calculation in your course material. All the rest is just the reading and just revising. It will be more interactive. So let's just look at this calculation as the last calculation that you deal with in this course. Calculation of genetic advance is also simple. All these calculations are not difficult because this is an introductory class. This is introduction to crop breeding. If you do crop breeding later, I don't think this university offers such, but you can decide to go and do your master's in breeding. Then you will know that this breeding is more complex than this. But this is just an introductory class. So that is why I'm trying to, for those of you that don't like mathematics and you are looking at, well, we said we are doing agriculture now. Why is mathematics in agric again? So you have to meet all this kind of simple calculation. So calculation of genetic advance. To calculate genetic advance, it's usually done after selection. These are the things that you need. Number one is the K value. You need your K value, which is called the selection differential. And that one is usually on a table. It's given on a table. So I will give you your K value. Like I said, I don't want to encourage you taking any paper to the exam. Don't take any paper and be telling your invigilator that we are using this table, this one, that one. No, don't take any paper to the exam hall. Then you need your VPH2, which is a phenotypic variance of the trait you are working on, and you need this heritability. All of you know now that the small h is the narrow sense heritability. So those are the three things that you need for this genetic advance. And the formula for it is this K, the constant. K is a constant that will be given to you. Square root of VPH, square root of your phenotypic variance times the narrow sense heritability value. So this is the formula. This one too, you have to get it into your brain, whatever means, just have it there. Because you cannot write anything on your body or write anything anywhere to enter the exam. Just get it inside your head by all means. By all means. Because you cannot tell if it will come out in the exam or not. I don't know. So this is the formula for genetic advance. You have your K square root VPH times H squared. That is formula. So this is an example. You have suppose a plant breeder started with a population with a mean. This is mean. No, this bar should be on top of this X. Mean of 8 inches tall. This sign is for inches. So his desire is to increase the mean height. His desire is working with a population of plants, maybe maize, for instance, and the maize is 8 inches tall, but he wants to increase the height. Okay, so from the population, he calculated the heritability to be, so you have your heritability factor here is 17% or 0.17. Like I said, heritability is always reported in percentage, and the phenotypic variance of 2 inches. So you have the value of your heritability, you have the value of your phenotypic variance, and then you have the value of your you have this mean value. So the breeder decided to select the top 10% of 100 individuals, 10% of 100 plants, calculate the genetic advance and what will be the mean in the next generation. You have your value, you have your genetic advance, you have the formula, K square root VPH times H square. For 10% of 100 plants, the K value on the table is inside your course material, but don't worry yourself with that table. I'll give you this value. I'll give you your K value. So the K value is 1.76. If you substitute all the numbers into the formula, you have your K value as 1.76 times, you have your heritability times, where is your, okay, I omitted something here, the phenotypic variance. I omitted the phenotypic variance here, sorry. But look at it here, it's better done here. You have 1.76 K value times, this is the phenotypic variance times, this is the heritability. So the answer you have here is 0.422. That means that the breeder will make a progress of 0.422. So the next generation will now be that 8 inches plus 0.422. That is 8.422. So the breeder is making a progress of 8.422. That is just what the question is saying. And the percentage improvement is 0.422 all over 8 inches times 100, which is 5.3% improvement. Any question? Any question? Is there anything you want me to clarify?
Yes, ma'am. I'm please, ma, if you can retake this portion, please.
I should take it again.
Yes.
Okay. Question is, suppose a plant breeder has started with a population with mean of 8 inches tall, and his desire is to increase the mean height. From the population, the heritability is 0.17 or 17%, and the phenotypic variance is 2 inches. Okay, is this phenotypic variance that is worrying you, right? So the breeder decided to select the 10% of 100 individual plants, calculate the genetic advance, and what will be the mean in the next generation.
Sorry, ma, what this X stand for? The mean X.
Mean. It stands for mean. X, there's supposed to be a bar on it. This bar that is shifting to the side supposed to be directly under. This is how
Okay.
write your mean. Let me show you on the whiteboard. This is how you write your mean. The mean is written like this. X with a bar on top. This is how you write your mean. So that is the best way that I can write it. So it says now that to calculate the genetic advance, and what will be the mean for the next generation. And I said this is your formula for genetic advance. Okay, K, the constant. This one is provided for you. The constant, if you go to your table, you will see the values there for 10%, for 20%, for, let me see if I can see it on your course material. There is, you see, share the link. This is a table here. You can see 1%, a fraction, the K value, 2%, blah, blah, blah. It goes on like that. Okay. So for 10% now, you can see the value. The K value is 1.76. So this is something like this that the table looks like. But like I said, this K value given to you, to be given to you. Nobody will tell you to go and find the K value. Given is a value on the table, just like you have your chi-square table. You have your K value table too. So you have your K value, then the phenotypic variance. Phenotypic variance, okay?
Yes. Square root of 2. It will be square root of 2.
Of 2, yeah?
Yes, which is 1.41.
This 2 should be here. Let me put it there. It's a mistake. Square root of 2. You can see. So you have your 1.76, which is the K value. You can see it in this formula here. Square root of 2, which is 1.41. And then you have your irritability. It was given to you in the question too, 0.17. So that is what you use to get 0.44422. So genetic advance will be 0.422. So what is the progress that the farmer would make if he's adding into the inches, the inches of his plants? That is 8. You can see 8 from the question here. The plant is 8 inches tall. So the genetic advance will be 8 plus 4.22. 0.422. That's why you get 8.422 here. And for the percentage improvement, that will be this same 0.422 all over that 8 times 100, which is 5.2, 5.3%. So you have a 5.3% improvement. So for your second assignment, you find the genetic advance for that assignment 1. Okoronko, do you have anything to say?
Yeah, I wanted to know if the man wants plant to be 8 meters short, how will he calculate it?
How will you
Calculate it. So is it that
Calculate
the
the 8 inches. That one is part of the question. There's nothing to calculate there. It's inside the question.
if he
It will
wants
be provided. Is that what?
said if he wants his bridge to be 8 inch short, 8 inch short, is he going to be minus instead of plus?
No.
Since the 8 inches tall is plus, which is improvement. Is he going to
If
be
it is
short?
improvement, is plus. Yes. It's an improvement.
If he wants it to be short.
Okay, if you want it to be short, then that one is not genetic advance anymore. It's not genetic advance. If it is advanced, then it has to be better. It has to be an improvement over the one before. Okay, so cannot be shorter. If it is shorter, then that means you're going back. Then the farmer does not want to go back. No farmer wants to go back in whatever he's doing. You want to advance. Hope you understand.
Yes,
So, so in this your assignment 1 now that I gave you, this one, you will see your heritability. You will get your narrow sense heritability from there. And then your K value, assuming your K value 2 is 1.76, and your phenotypic variance too, you get it from that question. So I want you to do the genetic advance, assuming your K value is 1.76. Hope that is clear. So
wait, sorry, ma. That means the narrow sense heritability will take it from the results of the first
yes, exactly.
Then we assume the K value is 1.670, 1.76.
Yes,
then
exactly.
we will take the mean in the mean as 8, right?
Yes.
Okay.
Take it as 8 too. And your phenotypic variance too is in that question.
Okay.
In your question.
All right. All right.
Any question? Nobody is talking again. Apart from that, this question, I want you to attempt it. Attempt it. That's why I said attempt this exercise. It's not by force, but it's just better. Attempt this exercise. I want to see what you will do. Attempt is attempt. So I want you to attempt this exercise. I want to see what you will do. So, any question? Any question? Don't worry, I'm not going to come with maths in the next class. Yes.
Ma, this last question now, you said the first assignment you gave us, so we have to take some values from the first assignment to attempt this last one, the one with 22 kg, the one you said we should look for the genetic advance.
Yes.
Yes. Thank you.
Let's find the genetic advance for the first assignment. Okay?
Yes,
and
ma.
I said you should attempt this one. Yes. Any other question? Is there any other question? Gloria, I can't hear your voice.
Good morning, ma.
How are you? Well done.
Fine. I did not have question until I attempt it first. But I do not ask question.
Attempt it. Let me see what you will do. Okay? In the absence of that, I think we can call it a day. So tomorrow is another day for CRP 303. For those of you doing CRP 303, we'll meet again tomorrow, same time. 10 o'clock is the time. In the absence of any question, I like to end this class and wish you the best. Please prepare. Your exams will start immediately after this facilitation. I think by next week you will be seeing your timetable or something. And this time around, it is not objective alone, like your 200 level. You people have grown too. So you should be able to write. All your exams this time is on pen on paper. Pen on paper. You will write, they will give you questions, you will attempt the questions. The first one is 30 marks and it's compulsory. Then you will attempt 2 other ones. So you're answering 3 questions out of 4. So please prepare.
I hope this crossing will not come up.
I don't know. Go and learn it. I don't know.
I'm learning that it takes a lot of time. So I don't want to sit in the exam.
That's why you have to practice. Practice is the one that makes perfect. You cannot know it overnight, but you keep practicing till you know it. Keep practicing.
Yes, ma'am.
So I don't know the question that will come out. That one is in the hand of DEA. I'm not DEA. I've done my work. So they will.
I hope they will. I hope they will, because
No, tri-hybrid. No, I'm not. Tri-hybrid is very, very cumbersome. I'm not bringing out that tri-hybrid. I told you to attempt it. I told you to attempt tri-hybrid, so I'm not bringing that one out in the exam. Tri-hybrid cannot come out in the exam. I just want you to attempt it and know. So
please, now why a big exam for us? Is it the one teaching us?
Who?
Please, the person that is speaking, you send the question, another person not speak for us.
Yes, I sent a question, another person will mark it. I'm not the one marking it too.
How will you know what we are doing is correct or
not? There's a marking guide now. It's not just marking, he has a marking guide in his hand.
That will not miss.
He has a marking guide. You know, in Open University, they want transparency. They don't even want you to have any contact with your lecturer. So it will not be as if, it's because this lecturer likes glory-headed space, so that is why she gave her 100/100 and does not like Farouk's face. So we don't want anything like that. So we want it to be as transparent as possible. We are not involved in we just teach, we set the questions. We are not involved in, you don't even see any lecturer in the invigilating. Have you seen any lecturer in invigilating before? Maybe just once or twice or something. They are not usually in study center. We are not in the study center, we are in the headquarters. Right now, as I'm speaking to you, I'm in Kaduna now. I'm in Kaduna.
Some question.
So, when they will mark too, we don't know those that are marking. That's why we always say, come to class. The best I can do for you now is to teach you and tell you to ask me questions. Ask me questions, I'll answer them to my best capability, and then you go for your exams. That's how it operates here. Any other question?
Are you the one setting?
Am I?
Are you the one setting the exam questions?
I said, yeah, I'm setting it. Yes, I'm the one setting it. I'm the one that set the exam questions.
So not the questions.
But I set many of them. I set many. That's what they told us to set. They don't want us to know the ones they will give you. So they said set many and submit. It's they that will now choose the one that they will give you.
Okay.
That's the way it works. They will choose. We don't know. My own is just to set the question. They will choose whatever they want to give you.
Okay.
So that's why I'm trying to rush and cover up everything so that it will not be like, this one that came out, she did not teach us, so we don't know anything about it. That's why I'm trying to.
Ma.
Yes, please.
Yes, concerning the exam question again, because it happened last semester in my 200 level. The question they
Okay.
said for us in one of the courses were not what were in the course material. So
Really? What course was that?
it has to be not I don't think it's part of that course. My colleagues too can testify to it.
One of them.
What is the name of the course?
Forgot it. Glory, do you remember the name of that course?
I remember one of my own.
Oh, remember, just forget about it. We have done it. We have done it. We have moved. Forget about that one.
Yes, I know. So that maybe the lecturers can help us.
You guys should not to go back to the 100 level, please.
We are doing.
It's only 2 years that I have. If you want to answer any question, you raise up your hand.
Ma, 100 level and 200 level, especially first semester, the experience was terrible because I can remember very well. Practical biology. Sorry, by 192.
192.
It was not about biology at all. The actual statistic.
By 192.
Statistic, yes.
So who did you complain to now?
Who do we complain to? Nobody, because nobody will even answer us. You took your exam, either you fail or you get E. Then there's nobody to take your complaint to. They give us statistics. I know that particular discouraged one of my friends.
See, most of you don't have a good rapport with your study manager. Your study center manager is the person you should lodge your complaint to. They are the ones that will bring it to the headquarters. This system is not a system that you cannot do anybody's work for anybody. Everybody, as we all are, we all have our schedule of duty. Now you are telling me I don't have
the working system, especially in the Abuja here, it's not something that somebody will just come on. You will see that as if they want to give you life, especially those on a non-academic staff. It's as if you and them are fighting or you are quarreling about something.
Non-academic staff is like that everywhere, even in conventional university. When we were in the university then, non-academic staff, if you want to pay your school fees receipt, just to give school fees receipt, they are a problem. They are like that everywhere. And that is, you should understand why. Because most of them know that as you are educated and that, somehow you are going to be better off. So they just have a way of creating problem. So that is why when you take your complaint, if they are not answering, take it to another quarter, another higher quarter. So nobody will gain anything from not letting anybody graduate. So you have to make sure you are the one paying your money. You have to make sure you are getting the best of it. Take your complaint. If somebody is not answering you, take it somewhere. Even try and cause a scene there. Then they will know, because the headquarters must not know that this person has a complaint and you are not attending to it. So even if they don't answer, you can cause a scene there and try to tell them that you don't understand what is going on. You should explain. So you have to do it by all means. Yes, Gloria, what do you want to say?
When we take our complaint, like now, e-ticket, they have given us e-ticket to complain. I want to do my complaint on e-ticket, send my TMA, some of the thing I got a low score with even the question I choose, with some question and options that are not correct. The highest they could tell me is that all the options are correct, whereas they are not correct. I have no say.
Did you snap those questions there for them? Just like you snapped it to me, did you snap it to them like that?
No, I did not snap it to them.
They have to see evidence.
So where did they not check to? I thought they went to my course page and checked, and I come back and tell you.
What they will tell you that you are not a serious student. I remember last semester, GST 204. I submitted my question to be very, very to the best of my knowledge, right? But they marked me wrong. thank God I submitted. I screenshotted all the questions. I submit to the this thing, the support ticket that I read. Nobody answered me. Nobody answered me anything. It was later that I found out that the particular GST, that GST 204, they use 3 different course materials to set the question. 3 different course materials. Even the main course material that we're supposed to get the answer from, the course material was about 207 pages, and you now bring 2 other to join this. It is not fair at all.
I mean 204, something like that.
In fact, I don't even know what to say.
3 different course material to do that.
We'll still graduate, but just grace.
Just do your best. Do your best. It is well. For all these complaints, I just wish there's a place where you people used to do students. What do they do every semester? I used to do like a. We used to meet before the year exam, before this facilitation starts now, like an orientation. You don't do that.
Our open university, big girl and big boys plenty. Like Gloria here, you know, they say if not, you go to your house. So where you go? Why you want?
Yeah, I will try my own best and try and complain.
Yes, you really try. Even the word of encouragement, you try. You try.
Thank you, ma'am.
We know that you are not part of that system.
It is well.
Thank you, ma'am. I'm not part of them. God bless you.
Thank you.
Well, have a nice day.
I you too.
Okay.
Thank you.